Hello everyone, my name is Xueqiang Wang. It's my great pleasure today to introduce our work where we constructed a pan-genome atlas for diploid cotton species, shedding new light on the genomic insights into the evolution of diploid cottons. Exploring the diversity of gossypium plants, particularly the evolutionary relationships among different cotton species, has long been a focal point of scientific inquiry. In 2023, Professor Fang Lu's team illuminated through genomic analyses that the rapid adaptive radiation in gossypium plants is shaped by widespread incomplete sorting and gene flow, emphasizing the importance of high-quality genome assemblies in deciphering the evolutionary history of the cotton genus. Preceding this in 2022, academician Xueqiang Wang's team constructed a pan-genome incorporating 10 representative diploid cotton genomes, uncovering correlations between transposon genome expansions and innovations in chromatin structure. Despite significant advancements, the pan-genome for all species harboring the D genome within the gossypium genus remains unconstructed, and sequence alignments focusing on nectar gland genes in diploid cotton species have yet to be accomplished. To address these gaps, we are building a comprehensive pan-genome encompassing all D-genome cotton species and completed sequence comparisons of nectar gland genes in diploid cottons. Highlights: we selected 22 representative diploid cotton species to construct a high-quality super pan-genome. Through in-depth exploration, we uncovered adaptive evolutionary features among gossypium species, elucidating the intricate genetic mechanisms underlying species differentiation and environmental adaptation. Detailed analysis of the pan-genome allowed us to identify 321 structural variant hotspots, with 90 genes directly implicated in the initiation and elongation processes of cotton fiber development. Addressing the unique phenomenon of nectar gland absence in Gossypium klotskianum and Gossypium trilobum, we pinpointed a specific 444 BP deletion in the promoter region of the Guan gene, providing a scientific explanation for this long-standing biological puzzle. The most TEs were found in G. rotundifolium, K2, and the fewest in wild relative Xueqiang. The cultivated diploid cotton diverged from the wild diploid cotton species about 5.45 Maya, and the divergence time between diploid cotton species and its wild relative G. Xueqiang was about 10.13 Maya. Collinearity blocks between assembly genomes were identified, and 13 collinearity blocks between G. herbaceum, A1, and G. arboreum, A2, were disordered on different corresponding genomes. Huge chromosome inversions may drive species formation and diversity. The pan-genome contains 67,807 genes, including 22,384 core, 34,093 variable, and 11,330 specific genes. KEGG pathway and GO enrichment analysis of core genes showed terms related to growth and development of cotton. Specific genes were enriched for different terms, revealing variations in characteristics of different cotton species. Results showed differences in total number and types of SVs across cotton species. Repeat contraction was most common and deletion least common. Unevenly distributed SVs were identified in 321 SV hotspot regions, including 90 genes associated with fiber initiation and/or elongation. No foliar nectary was found in G. gossipioides, D6, G. schwendemanni, D11, and G. tomentosum, AD3, the allotetraploid cotton species. Comparative genomic analysis of GON expression revealed no expression in D6 and D11, suggesting a lack of function of GON in preventing foliar nectary development in these two wild diploid species. Sequence analysis of the GON promoter sequences showed a large deletion, for 44 BP fragment, in the promoter sequences of GON from D6 and D11 species compared to other diploid cotton species with foliar nectaries. Summary: constructed a high-quality super pan-genome of 22 diploid cotton species and investigated their adaptive evolution, identified structural variations and hotspot regions associated with fiber initiation and/or elongation, investigated the absence of foliar nectary in G. gossipioides and G. schwendemanni, and identified the deletion in the promoter sequence of GON as the cause.